Alrighty, so I'm Lucas, and that's Grace, and we're going to be talking to you guys today about our assigned and our picked paper for population genomics. And our assigned paper is going to be about Neanderthals and their ancestry as it relates to present-day humans. So, pretty much the basis of the paper is like modern-day humans are a mix of a bunch of somewhat deleterious Neanderthal alleles with our genomics that we have from today. And the Neanderthals are going to be the non-African ancestry. So people that I guess came from pretty much like right outside Africa. So they were able to detect certain Neanderthal alleles at specific loci, and that's how they were able to kind of figure out who had these Neanderthal alleles by looking at it. And they mapped out all 23 chromosomes to do so, which I'll show you on the next slide. And they used a CRF, which is a conditional random field, and they used three features to kind of establish this to find genetic variation as it deals with Neanderthals. The first one is the allelic patterns at a single nucleotide polymorphism. Another one is going to be high sequence divergence of non-African haplotypes to all the YRIs, but low divergence from Neanderthals. And the YRIs, they're just using those as a basis for people that don't have Neanderthal ancestry. And the last one is going to be length of the specific haplotype. They're looking for about 0.05, I think it was centimorgans. And this is consistent with interbreeding that happened 37 to 86,000 years ago. So this is going to be the pictures and the maps of like all the chromosomes, all 23. So you have the 22 autosomal and the one X-linked chromosome, or the one X chromosome that they're going to map out. The little black dot in all the pictures is going to be the centromeres, and they kind of just show you the specific regions where these certain alleles are more frequent at. So on the left side of the graph, there's the 0 to 0.1. That's showing like 0.1% versus the length of the actual chromosome. And the red is going to be the East Asian populations versus the green, I believe, is the European population. So they're kind of showing the differences between who has more alleles on which chromosome. So a little more into the ancestry and how they did it. They use for their CRF, they use a 1,000 genome project preparation, or for part of their preparation, which is a giant database started back in 2008. And they use it to pretty much map genetic variation in humans. So it's just this giant database that you can go on and use it for, I guess if you want to do like hominin studies and link certain alleles and stuff like that for the human genome. But they use this for the European and East Asian populations, and they identified alleles from Neanderthals that affected the following, mostly diseases, but it's going to be smoking, lupus, biliary cirrhosis, Crohn's disease, optic disc size, and also type 2 diabetes. And I think it's in the next table on the next slide that I'll show you, but I believe one of the populations is way more prevalent for having type 2 diabetes. So that was something that they were able to map out by finding these Neanderthal alleles, which I thought was pretty cool. And they screened for haplotypes in the 1000 Genome Project by using the Altai Neanderthal genome of 52-fold average, and they used a 6' consensus to determine certain ancestral alleles. So obviously the hominins came from primates, so they use, I guess, 6 different ones to try and determine certain ancestral alleles that would be present in Neanderthals. And there was 176 of the YRIs, which, like I said, they use as a basis for people that weren't supposed to have Neanderthal ancestry, or non-Africans that don't have Neanderthal ancestry. And they found that significantly enriched alleles for people with these Neanderthal leftover alleles that are still present are going to be like keratin filament formation alleles. So that's why in the picture that you saw on the first slide, he was really hairy. Um, but they thought that these alleles affecting like hair and skin may have helped them to adapt to non-African environments. And yeah, so this next table is going to show talking about that type 2 diabetes and all the diseases, the certain phenotypes that would be expressed in the presence of the Neanderthal derived alleles. and they used the GWAS for this. But it'll show you like the derived alleles, kind of like where they are, and also the European versus the East Asians. It's showing you the percent frequency for certain derived alleles and what they would express as the phenotype if they were to have that. So the East Asians are way higher in type 2 diabetes than the Europeans. And some findings from their study were that multiple derived alleles showed risk for diseases. So as you saw in the last slide, certain Neanderthal alleles are going to code for, I guess, certain diseases that we see nowadays in humans. So this Neanderthal DNA is still shaping today's modern bio because we're still trying to figure out kind of the direct causes or how to combat certain diseases like this. So regions on chromosomes that lack Neanderthals are going to be rich in other genes. So natural selection is actually selecting for the removal of a lot of these Neanderthal alleles. And certain allelic combos have caused increased mortality in males. So that's why a lot of these deleterious alleles are getting booted out. And they found that the proportion of the genome confidently from Neanderthal ancestry in the East Asian populations that they were looking at was about 1.38% versus the Europeans was 1.15%. So that might be why you see more expression of like certain diseases in the Eastern Asian populations than we saw in the European ones. And this is also consistent with previous reports that showed an increased Neanderthal ancestry in the Eastern Asian populations. So they have been pretty common in having more Neanderthal in them. And more findings, pretty much the interbreeding led to that male sterility. Neanderthal genomes more prevalent in the Eastern Asian populations, which I mentioned. And it might have been the reason why they're getting booted out is kind of like a snowball effect. All these accumulations of bad genes, our bodies are, I guess, able to somehow detect it and try and get rid of them all. But it's important to explore that if this happened in other interbreeding events throughout the hominin lineage, so that way we can kind of like find out more information about it.
Perfect. And then the selected paper we got was an investigation on genetic history of Japanese populations of 3 starfish and their DNA sequences. So the crown of thorns starfish this is kind of a little bit of an overview on them. They're located in the Indo-Pacific and coral reefs, and they have many outbreaks that invest onto the coral reefs that are implementing how bad the coral reefs are growing. So they are a little bit of an invasive species. And so with these outbreaks, people remove them from the coral, and we were looking at 2 starfish. So the Pacific sea star and then the blue starfish were used as controls to see exactly how this crown of thorns starfish population is being impacted by the removal of these species. And they looked at the locations, these tropical Japanese islands. They're subtropical and southwestern. I can't pronounce it, but if you look down at table 1, you can look at the locations and the total number, the approximate latitude, and which year these 3 starfish were collected at. So the research continued. In previous studies, they used mitochondrial DNA and microsatellite loci to report a genetically homogenized pattern of crown of thorns outbreaks. So they used different approaches in using statistical analysis and different ways of contracting that DNA. But in this study, they used a different technical improvement and used sequence. Instead of doing a full mitochondrial DNA sequence, they used the 2 other starfish as well. And this was a sequence comparison for single genes, and this would not produce useful result for crown of thorns starfish. Hence why they did the full mitochondrial DNA sequence. And then the pictures here, the first one is mitochondrial, not the mitochondrial, the crown of thorns starfish, the blue starfish, and then the northern Pacific starfish. So pretty much the genomic DNA was extracted from all 76 specimens of the 3 different species, and it came through their feet. So with the whole mitochondrial genome sequence, the divergence events was calibrated into a tree using real time. So that is what the graph on the right is, and the arrow shows the separation of these species 185 million years ago. This is important because figuring out when the divergence time of these species in putting it into a data set for all of these species really represents how exactly the divergence of the population occurred. They also analyzed population structures using a model-free approach, which is important as well because that is what I'll be showing in the next slides. So they used a long PCR method. So this was the complete mitochondrial genome sequence of each, the Northern Pacific sea star, the blue sea star, and the crown of thorns sea star. I'm not going to try to pronounce these because I will mispronounce them, but the first one's the Northern Pacific sea star. That's their full mitochondrial genome, which is important to see the difference in their genomic structure versus how similar they are in their structures as well. So the main findings was the Pacific sea star. The A is going to be the adult starfish. The B is going to be the location where the samples were collected. So obviously in the Japanese islands. And then the Pacific sea star showed a divergence of 2 species at 4.7 million years ago. And then the letter D shows how the population supporting pretty much how the tree of 4.7 million years ago and those 2 specific species. And then the blue sea star, there's a picture of the blue sea star, their location in the Japanese islands, as well as their tree, noting that it was 6.8 million years ago when these 2 populations were diverging. And then D supports C, showing the population divergence in those different species in their genes. And then the crown of thorns, this is the most important part. So those were the 2 controls of the group to figure out, see exactly how these sea stars are being impacted by population genetics. But this one, so they noticed that 5 different populations diverged 0.7 million years ago. And obviously, if you look at number D, you can see group 1, group 2, group 3, group 4, and group 5. Those are all different species that had diverged 0.7 million years ago. And that's extremely important, which I'll explain in the next slide. So this is important because both the Pacific sea star and the blue sea star showed 2 populations in the Japanese island region diverging like 6.8 million years ago for the blue sea star and then 4.7 million years ago. And there's been no genetic exchanges since for the Pacific sea star. And this suggests the presence of 2 cryptic species in these Japanese islands for the blue sea star. But the crown of thorns starfish showed at least 5 lineages since their divergence. So this represents a possibility of repetitive genetic bottlenecks caused by the outbreaks. So when people remove them, they have a little amount of population left, so they're reproducing and their genes are being transferred through bottleneck genetics and that experience. So therefore, they're going to have more lineages and more species versus the other 2, which are not being removed by anthropogenic causes. So the comparisons, there's divergence in both species. So when alleles, there's obviously the allelic divergence for both, and they are both prominent in divergence between species. So for example, mine's starfish, his is going to be in different regions for, both papers, and yeah, both papers included population genomics as well, which is the main cause of this presentation, and they were both location based. So his was in East Asia and Europe, and mine was in Japanese islands. They also used statistical analyses to see DNA sequences, which is also important.
And then some differences that we had were going to be pretty much like how versus when certain alleles diverge between populations. Mine was more of how, and hers was more of dealing when, and obviously starfish versus humans. But our sign was about human divergence. Like I said, pretty much just above, but the selected paper is going to be about the starfish population specifically. And our selected paper also focused a lot on the many statistical systems, excuse me, can't speak randomly, to determine the conclusions, versus the assigned paper was more of like a research base and kind of like a field study in a way, because they had like direct contact with the organism. So
yep. And then those are our references.
All right.
Stop recording.